Welcome, everybody. My name is Steve McConaughey, and I am the Executive Director of the Global Nerve Foundation. I'm very excited today for our abstract spotlight to be speaking to Dr. Justin Burrell from the University of Pennsylvania. Dr. Burrell is a postdoc fellow and works in the Lay and the Cullen labs at the University of Pennsylvania, with a focus of his research being on peripheral nerve health and peripheral nerve regeneration. So obviously it fits very well into the mission of the GNF. They had some exciting results in their recent publications, and Dr. Burrell is here to share them with us today. So thank you for being here, Dr. Burrell, and I'll turn it over to you.
Thanks, Stephen. It's my pleasure to be here. So this is, as you mentioned, this is some exciting research we've been working on the last couple of years. It's actually a collaboration between myself, the Cullen lab, and the Cardozo lab in Mount Sinai and the VA. And so this is a really unique project where we're focusing on an area that previously has been understudied, and that's the relationship between the capacity for regeneration due to the stabilization of acetylcholine receptors and muscle. So basically, muscle dies, and we need to find a way to prevent it from atrophying after a nerve injury, and that's what we've been studying here. And so, as your audience knows, the peripheral nerve injuries are devastating, and they cause usually semi-permanent damage to leading to sensory and motor deficits. It can happen in a lot of different cases, to trauma or tumor resection, or sometimes even iatrogenic injury. And currently, the gold standard strategies for these injuries are autografts. So basically, suture repairs using either direct nerve repairs or autografts. And then there's other approaches such as acellular nerve allografts and conduits. But the important takeaway from these strategies are they're just focusing on regeneration, getting axons from point A to point B. They don't address the underlying issue that the muscle starts atrophying after nerve injury, and it's unstoppable. In fact, that's what we've thought for a long time. And this catastrophic axon degeneration, or sometimes it's referred to as Burrell degeneration, occurs almost immediately after axon injury. You start the axon starts breaking down and going through fragmentation, and it leads to the muscle. It leads to the muscle, so the axons degenerate, and then the muscle without axon contact starts atrophying, and then axons from the injury site have to then regenerate and reach the muscle target in order to get functional recovery back. And this is slow. It takes about 1 to 2 millimeters a day. This is why some patients will not have any functional recovery for sometimes months to years after a nerve injury and repair. So as I mentioned, the main focus that previous studies have looked into has been trying to accelerate regeneration. And ultimately, the ceiling for accelerate regeneration, but ultimately the ceiling for functionality is fairly limited without addressing this atrophy problem. And that's what we looked at here. And we've been studying a first-in-class pharmacological approach that stops this denervation-induced atrophy process. And in this study, we investigated boldine. It's an orally derived alkaloid, and it has been previously shown by the Cardozo lab and others to have interesting properties that prevent this atrophy process from happening due to by blocking connexin hemichannels. It's basically a mechanism that allows for calcium waves to occur and limits and basically locks up the muscles and causes further damage. But ultimately, in this study, we were looking at whether or not oral treatment of boldine could prevent this muscle atrophy from occurring after nerve injury, and if so, does that raise the ceiling for future functionality after delayed nerve repair? And indeed, what we found early on was we did a nerve injury. So in a rat nerve injury model, we cut the nerve and we allowed the axons to degenerate and the muscles to denervate. And after treatment every single day for up to 4 weeks long, we looked at the muscle recovery, and we found that there was the electrical activity persisted despite there being no axon connections. The electrical response persisted for up to 2 weeks, and there's a trend for some 4-week response as well. But mostly we found this interesting early acute response. We also looked at the muscle and found As expected, the connexin expression decreased with boldine treatment, but notably, it also was the muscle fibers were also larger and more robust following treatment. Means that the atrophy process was delayed and allowing for larger fibers to persist. Later on, after treatment, so after deinnervation following treatment, we then looked at reinnervation by doing a delayed nerve repair. Basically, we did a direct nerve repair and studied the muscles and nerves afterwards. So we found that the electrical activity, so the functionality in the muscle, was greater at 6 weeks after delayed nerve repair following boldine treatment. And also we also found that the axons were more mature and myelinated in this cohort as well. So this is showing us that the muscle was able to receive axons and also be and the boldine was able to facilitate maturation as well. We also then looked at the acetylcholine receptors and found that there was a greater number of them in the muscle. So by taking boldine every single day, basically there's more availability of the acetylcholine receptors, which is what the axons plug into in order to have functionality over time. And that can be shown here in panel D, which is the reinnervation of neuromuscular junctions over time. So in conclusion, what we found was that boldine treatment prevents this deinnervation-induced muscle atrophy, ultimately raising the ceiling for functional recovery following delayed nerve repair. We are currently testing this in large animal study as well. We're fortunate we just received funding for this by the VA for large animals safety and efficacy. And future clinical studies will determine if this strategy can be used prophylactically for nerve injury. So you can envision maybe being used in cases where we're not sure how severe the nerve injury is. And that's an exciting opportunity for us as well. With that, I would like to acknowledge my collaborators, as I mentioned, Cullen Lab, the Lay Lab, Cardozo Lab, and others.
Excellent. Thank you, Dr. Burrell. So some very exciting results. Looks like a kind of a novel treatment. Do you know, or are you aware, is there any preliminary work or any precedence of using boldine in humans and its possible efficacy as you kind of work your way from small animals to large animals and then hopefully clinical trials?
Yeah, that's a really good question. So one of the interesting things about boldine, it's been used for centuries now by indigenous cultures throughout Chile and China, where they've been steeping it in teas for centuries now. So
Oh, wow.
it's very interesting that it obviously hasn't been studied in any clinical experiments, but that's something that we're looking forward to do, is finding out what are the requirements that are needed in order to get therapeutic efficacy, and perhaps are there strategies that we can look into to improve delivery, maybe using some new bioengineering techniques.
So kind of more targeted delivery versus like the oral administration.
Exactly. The oral administration is pretty cool because it could be taken as a pill. We can envision this maybe being used prophylactically for. I'm sure as your audience knows, that the wait and see problem is one of the hardest challenges that patients face, right? And I was just talking with someone last night about this, where it's like they find out that they may have a nerve injury, but they have to wait 6 weeks to determine if they need surgery or not. Right. Hopefully they don't. Hopefully they recover, and they might get 80% recovery back, right? But the patients who need the surgery 6 weeks later, they might get 20% back because they needed early intervention and didn't have it. So it might be useful to have a prophylactic drug where effectively anyone that has a possible nerve injury is taking this drug, so that those patients who have 80% recovery, maybe they'll get 90, 95%. And the patients with 20%, maybe they'll get 40, 60, 80, who knows how much. But that's the area that we're excited about right now.
That is very exciting. So along those lines, when you're kind of talking prophylactically, did you guys see any other effects of the boldine, so other muscle health that weren't injured, or did you look at any of the like off-target effects of boldine for muscle health just in general in these animals that received it?
So those are good questions. So we did not see any adverse effects from the drug. Granted, this was in a rat study. We are going to be looking at that systematically in an upcoming porcine study. So in a large animal study, we'll be looking at the pharmacokinetics and looking for any side effects from the drug from different dosages that are needed. Because you're right, like, so if you're doing this in oral therapy, we had to do some pretty high dosages for the rats, obviously. And in humans, we definitely will have to need different requirements, but as you get higher in dosage, you get more problems, which is why one areas we're looking at is looking at trying to improve delivery, right? That's one thing we're looking at. But regarding other interesting findings, yeah, so we found that the drug does seem to have an effect on Schwann cells and axons. That was a little surprising, but as we dug into the research and we learned a little bit more about what the connexin hemichannels, how they affect the glial cells and the axons, it kind of made sense. It was interesting, and it's definitely a new target for us, like a therapeutic area for us to focus on. And so rather than speeding up regeneration, can we slow down degeneration? And you know, and that's future.
It's a very creative way. You're kind of looking at the other side of the coin, right? Like there's so much focus on getting the axon there as quickly as possible, but making sure that something is still there for when the axon arrives and being able to maintain that function is very interesting and again very creative. So thank you so much for sharing the work with us. We're excited to see where it goes, and all the best to you and your team.
Likewise, thank you so much for the privilege to talk with you.
Thank you.
Yep.